Here we have a sagittal section through one testis. And as you will notice, there's a lot of little ducts, a lot of little tubes found in the testis. We're not going to learn all of them, but we're going to learn 3 of them on this picture. The first are the seminiferous tubules. The seminiferous tubules are found all throughout the testis. You can see that there's a number of kind of sets of these tubules. These seminiferous tubules are the site of spermatogenesis. So found inside these little tubes are the cells undergoing meiosis in order to form sperm. So meiosis is occurring in the seminiferous tubules to produce sperm. When those sperm are produced, they still have to mature a bit. So that if you follow the seminiferous tubules, they will be transported by these little straight tubules, then into this rete testis, and then into these tubules here, or these ducts, which are known collectively as the epididymis. So that's the second most important site we see here. First is the seminiferous tubules, then the epididymis. And the epididymis is where these sperm are allowed to mature and be stored in these ducts. Then, from the epididymis, they will travel into the vas deferens, which brings the sperm out of the scrotum, through the spermatic cord, and into the pelvic cavity. So in this picture, I would highlight seminiferous tubules and what they do, epididymis, what occurs there, and the vas deferens. The next thing we're going to do is learn about how spermatogenesis occurs inside these little tubules. What we have here is a small little transverse section of just part of the seminiferous tubule. This would actually be kind of like the wall of the tube, and this is the inside. This is the lumen of the seminiferous tubule. And you'll notice the lumen of this tubule is full of cells. Most of these cells that you see here, at least the ones with the kind of the purple nucleus, those are all spermatogenic cells. Spermatogenic cells are cells that are going to become sperm one day. But they're not the only cells found in the seminiferous tubules. There's another cell type known as Sertoli cells or sustanacular cells. Those are the ones with kind of the pink nuclei. So you'll see one here, there's another one here, there's another one here, but you don't see the nucleus of this one. These Sertoli cells are a supporting cell. They help to support the developing spermatogenic cells. They help nourish them, they help support them. You'll notice that they're kind of surrounding a lot of the spermatogenic cells. They prepare them. They make the cells become smaller and smaller because sperm are very small cells. So they help that kind of development into a sperm. And another important and interesting function is they form a barrier known as the blood-testis barrier. What it is it's a physical barrier between the developing sperm and the bloodstream. And the reason why we need a barrier here is because your immune system is found in the blood, and if you're a male, you don't want that immune system to see all these different sperm cells developing. Because remember, one of the most important properties of meiosis is the formation of genetically diverse gametes. So these sperm are very different than all the other cells of the body because of that genetic variation. So the immune system would not recognize these cells as being good guys because the immune system is trained to only know what our somatic cells look like, our body cells. Once we start changing the genetics through meiosis, those cells look quite a bit different. So you don't want the immune system to notice that there are some cells that look a little different because the immune system kills cells that look different. And that's actually one cause of male infertility, is the breakdown of this blood-testis barrier. There's another cell type shown out here, kind of with the green look to them. Those are known as Leydig cells or interstitial cells. Interstitial because they're actually in between these seminiferous tubules. They're not found inside the lumen of the tubule. And their job is to produce testosterone. So they're basically an endocrine cell. Then you've got all these different stages of spermatogenesis. There's an original stem cell, which is diploid, 2N meaning 46 chromosomes. Those spermatogonium have to divide by mitosis. So there's a lot of mitosis that occurs here as well. But at some point, that cell undergoes meiosis, and it becomes a primary spermatocyte. Then that divides into 2 secondary spermatocytes. And then the 2 secondary spermatocytes are going to divide into 4 spermatids. So the process of spermatogenesis produces 4 spermatids. A spermatid is a very immature sperm. It's not ready yet. It has to further develop into a sperm cell or spermatozoa. This is a little more detailed drawing showing spermatogenesis, but you'll notice here is what I mentioned before, in that spermatogonia have to divide by mitosis. Males produce, on average, 300 million sperm a day. So you have to keep repopulating the cells that are basically precursors to sperm. Those are these stem cells, the spermatogonium. But you'll notice this cell divides into these 2. This cell is probably going to divide into 2. But this guy here has decided to undergo meiosis. We call that kind of decision-making differentiation. So the spermatogonium differentiates into a primary spermatocyte. That guy undergoes meiosis 1. So meiosis comes in 2 sets, 2 divisions. The first one, from this guy, primary to secondary, occurs during meiosis 1. And that's when a lot of the genetic diversity and variation occurs. You get, one event is known as crossing over. That's where genes get swapped between chromosomes. So like there'll be a chromosome 1 and another chromosome 1, but they'll start trading DNA to produce completely different chromosome 1s. So a lot of that genetic variation occurs in meiosis 1. Then the 2 secondary spermatocytes will then divide during meiosis 2 to form 4 spermatids, and they're all kind of connected to one another with these cytoplasmic bridges. So there's no way these guys are ready. So they have to undergo another process called spermiogenesis. That is when you take the spermatids and they further develop into sperm cells. So notice you get 4 haploid sperm for every 1 primary spermatocyte. Those sperm look like this, where there's a tail portion that's divided into a middle, principal, and end piece. Inside the middle piece, there's a lot of mitochondria, because this guy has to produce a lot of energy to drive this tail, or the flagellum. The nucleus is found in the head region, and it is covered or surrounded by a structure called the acrosome. The acrosome contains a lot of enzymes that are required during fertilization. So this acrosome will release these enzymes, and that helps the sperm fertilize the egg. When sperm enter a female, it takes them a while to, what we say, become capacitated. So basically, kind of shake out the cobwebs from the process of being delivered into the female. Then it kind of has to, take hours before it's ready to swim, if you will. So there's a time period there where the sperm really are incapacitated when they enter the female, and it takes about 7 hours to become capacitated. Then it depends on who you talk to. I think your book says it's got about 48 hours to fertilize. Others suggest, it's got up to about 5 days to fertilize. So most suggest that sperm can be in a female 4 or 5 days and still have the ability to fertilize an egg. And typically it's believed that to increase the chances of fertilization, sperm should be in the female before ovulation occurs. This last slide goes over the hormones of the male reproductive system. And many of these hormones we've done in the past. This is a list of the 5 hormones that you should know. Some of these, like the first 3, are also found functioning during the female reproductive system. GnRH stands for gonadotropin releasing hormone, because these are the gonadotropins, FSH and LH. So gonadotropin releasing hormone causes the release, the synthesis and secretion of FSH and LH. And that starts at puberty. So the initiation of puberty is when GnRH from the hypothalamus stimulates the release of FSH and LH. FSH stimulates spermatogenesis. So you'll notice FSH is going here. This is supposed to represent spermatogenic cells in the seminiferous tubules. So FSH directly stimulates the production of sperm. LH stimulates Leydig cells or interstitial cells to produce testosterone. Then testosterone also stimulates spermatogenic cells to develop. So both FSH and testosterone stimulate spermatogenesis. Testosterone also gets converted into a slightly different form called dihydrotestosterone, and together those 2 hormones bring about the male pattern of development. They cause the enlargement of male sex organs and expression of male secondary sex characteristics that occur at puberty. So the voice lowers, muscle mass increases, bone mass increases. Males distribute their fat in different locations than females based on testosterone. So all those male characteristics are due to testosterone and dihydrotestosterone. They also are anabolic. They stimulate protein synthesis. So those are the hormones in males. Females are going to have a couple extra hormones. So we want to really nail the male hormones because they're fairly easy when compared to female hormones.